Okay, let's go ahead and get started. Hi everyone. Thank you so much for joining our Meet the Expert webinar today. I'm really excited to have Dr. Kevin Qian here today to talk about his new findings in brown adipose tissue metabolism. Before I pass it over to Dr. Qian, I just want to share a few company slides to introduce Proteintech. So Proteintech is a leading manufacturer of antibodies, proteins, and immunoassays across several different research areas. Its portfolio comprises of three core product lines. So we have our Proteintech branded antibodies and immunoassays. So we have antibodies against over 13,000 targets. We also offer antibody labeling kits, ELISA kits, IHC kits, and cell separation kits. We also have our humankind branded bioactive proteins, and all our proteins are expressed in human HEC293 cells so that you can be confident that they have native folding, post-translational modifications, and glycosylation. And lastly, we have our Chromatech branded reagents that are based on alpaca nanobodies. So the product lines all share common values under the main Proteintech brand. So you can be confident they have high quality and undergo a thorough validation process for scientific excellence. But they all address its customers in a separate and specialized manner. I also just want to take a moment to celebrate the success of our customers because our customers' success is also our success. And we're really proud that Proteintech products have been cited in over 190,000 publications worldwide. We're also really proud that we were the winners of the CiteAb Researchers' Choice Award from CiteAb last year, showing that our customers really do value our products. So with that, I want to introduce our speaker today, Dr. Kevin Qian. So Dr. Qian is currently an MD PhD student in the UCLA Caltech Medical Scientist Training Program. He completed his undergraduate studies at Yale College in 2014 and his PhD at UCLA in 2021 in the laboratory of Peter Tontonas. He will receive his MD from UCLA in June of 2023, and he plans to pursue further clinical and research training in internal medicine and endocrinology. So with that, I'll go ahead and pass it off to Dr. Qian. If at any point you guys do have any questions for him, please feel free to input those into the chat or the Q&A box. So thank you so much, Dr. Qian, for being here today, and I'll go ahead and pass it off to you.
Thanks so much for the invitation and the kind introduction. Let me get my screen shared here. Let's see. Okay, let me see if I can hide this. Okay, great. So today I'm going to share a little bit about my thesis research from the Tontonas lab at UCLA. The title of my talk is Droplet Like It's Called, Calcitonin 3 Beta Enforces Multilocularity in Thermogenic Adipocytes. As you might be able to tell from the title slide, I'm going to be talking a little bit about the unique lipid droplet biology of brown adipocytes. A little bit of background. One of the areas of interest in the Tontonas lab is systemic energy balance, which is kind of the push and pull between energy intake and expenditure. Energy intake is primarily controlled by feeding behavior, whereas energy expenditure receives inputs from basal metabolism, physical activity, and a process of adaptive thermogenesis, which is essentially heat generation in response to diet and cold temperatures. And imbalance between energy intake and expenditure towards intake is underlying mechanism for a development of obesity, whereas energy imbalance towards expenditure can cause cachexia and adipose and muscle wasting. Adipose tissue plays a really interesting role in this balance because it can contribute to both sides of the equation. White adipose tissue is kind of the conventional fat tissue that you think about that stores energy that you take into the diet, whereas brown adipose tissue is a type of fat that expends energy through this adaptive thermogenesis process by generating heat in response to diet and cold temperatures. And interest in brown adipose tissue has really been revitalized in the past couple of decades due to the identification of brown adipose tissue in adult humans. Previously, it was thought to be restricted to kind of infant humans and rodents. And here you can see in 3 different papers in the New England Journal of Medicine in 2009, brown adipose tissue was identified as regions of high 18F-fluorodeoxyglucose PET uptake in a lean adult male exposed to a mild cold stress of 16 degrees Celsius. You can see that there's FDG uptake in the supraclavicular region, in axillary region, and mediastinum, corresponding to regions of thermogenic adipose tissue. And more recently, a retrospective analysis of tens of thousands of PET scans at Memorial Sloan Kettering in New York showed that the presence of brown adipose tissue in humans is associated with decreased incidence of type 2 diabetes, dyslipidemia, and heart disease, although causality obviously has not been established in humans. But in mice, brown adipose tissue has consistently been shown to have anti-obesity and anti-diabetic effects. So the differences between the different types of adipocytes, on the left here, you can see an H&E stain of white adipose tissue and a cartoon of a white adipocyte, and on the right for brown adipocytes. And some of the key differences are that white adipocytes have few mitochondria, whereas brown adipocytes have many mitochondria that are positive for a protein known as uncoupling protein 1 or UCP1. And this protein is what enables brown adipocytes to dissipate chemical energy and generate heat, whereas white adipocytes are specialized in storing and releasing energy. And what I'm going to be talking a little bit about today is that one of the other characteristic features of white adipocytes is that they're unilocular, meaning that they store their lipids in one large lipid droplet that seems to essentially take up the entirety of each cell. Whereas in brown adipocytes, lipid stores are organized into many small lipid droplets known as multilocular morphology. And there's a third type of adipocyte known as a beige adipocyte. This in many ways is similar to a brown adipocyte, but these kind of develop within white adipose tissue depots in response to cold. They're kind of recruited in response to the cold stimulus. And one interesting concept that you find in biology, but also in other fields, is form follows function, meaning that the form that something takes on in some way is optimized for the particular function that it might be carrying out. And conceptually, we've applied this to white and brown adipocytes in the sense that white adipocytes are unilocular. The one large lipid droplet is really efficient for storage of lipids, and also makes the lipid droplet surface very close to the plasma membrane of the cell and allows for efficient release into the circulation. Whereas thermogenic adipocytes are multilocular, containing many small lipid droplets, which increases the surface area relative to the volume and allows for access for lipolytic enzymes and mitochondria to mobilize and oxidize fats. So this has kind of maybe been assumed by many of us in the field, and I think we kind of set out to figure out the molecular underpinnings of the multilocular phenotype and more definitively show that this was advantageous for brown fat function. Just a brief aside, I'm going to talk a little bit about an abbreviated history of brown fat, which is essentially that brown fat was discovered by a physician, Gessner, in 1551 in hibernating animals. But it wasn't until 1960s and 70s that brown fat was discovered to be a thermogenic organ, that this was mediated by UCP1, and that it was involved in energy balance and obesity. So in the 4 centuries between its first description and discovering its actual function, there was really one, major characteristic of this tissue that was known, which is what it looked like under a microscope. This is a paper from 1923 showing axillary adipose tissue in an adult rat and also perirenal adipose tissue in a human infant. showing the characteristic multilocular morphology of thermogenic adipose tissue. Here you can even see it's these thermogenic adipocytes are mixed in with the white adipocytes that have the characteristic unilocular morphology. So this has really been known for a long time, and I don't think the molecular underpinnings of this difference has really been described. And interestingly, at the very end of this article, there's a line that says, until the nature of the structure is better understood, it would seem unwise to discuss the relative merits of the various terms applied to it. As you can see, it was frequently referred to as the hibernating gland at that point. And the last sentence says, possibly multilocular adipose tissue would be the least objectionable, suggesting that this was really the defining characteristic of this tissue that was kind of agreed upon at that point, at least until the actual function of the tissue is described much later. So the central question that my thesis research tried to contribute to answering is how and why do thermogenic adipocytes enforce a multilocular phenotype? This was actually not necessarily the question that we set out to study. This is just kind of where our research led us, and we were really excited by the findings. So this story starts with a novel gene/protein that we identified that was specifically expressed in adipocytes. So there's this gene with a gene symbol CLSTN3. This is a gene that was previously thought to be expressed exclusively in brain tissue. So here you can see cortex RNA sequencing data and exons 1 through 18 of this gene in reverse direction from right to left, and that each exon of this gene is expressed quite nicely in the cortex, corresponding to a promoter at the very beginning of this gene marked by H3K4 trimethylation. And we noticed that in brown adipose tissue, something very peculiar is happening, which is that there's actually a promoter within this intron all the way downstream, and it's producing this novel transcript containing this previously unannotated exon here, connected to the last two exons of the known CLSTN3 gene, exon 17 and 18. So what we essentially found here, just by looking at these browser tracks, is that hidden within this intron of the CLSTN3 gene, there's a brown adipose tissue specific promoter that leads to the production of a brown adipose tissue specific transcript with a previously unannotated exon that could have some interesting protein coding function. And to prove that this transcript was specific to brown adipose tissue, we did qPCR analysis in a panel of 15 different mouse tissues. And you can see here that when you design primers targeting the first two exons of the CLSTN3 gene, you find expression only in the brain. If you design primers targeting the last two exons, you see expression in brain and brown adipose tissue. And if you design primers targeting the brown adipose specific transcript, which is called CLSTN3B, you see expression primarily in brown adipose tissue, but also a little bit in the liver and white adipose tissue, notably not expressed at all in the brain. What else is interesting about this is that the brain protein CLSTN3 is 956 amino acids with a large N-terminal extracellular domain, a single pass transmembrane domain, and a short intracellular domain. And the brown adipose tissue protein, or at least putative protein, is missing this entire N-terminal extracellular domain and has essentially swapped it out for this novel, previously uncharacterized N-terminal domain. So I like to call this kind of like a nature's domain swapping experiment. Essentially, this part of the protein has stayed on in the brown adipose tissue, but this extracellular part has been swapped out for this uncharacterized domain. I'm going to talk a little bit about Proteintech, not because Proteintech asked me to talk about them, but because they did actually play a very important part in this story. Essentially, we wanted to prove that this protein actually exists endogenously in brown adipocytes. And in order to do that, we needed to find an antibody that recognized the C terminus of the known calcitonin 3 protein. And at the time, all published antibodies were targeting the N terminus of the protein, which wouldn't really be useful for studying brown adipose tissue. Except there was a previously, I guess, uncharacterized, unverified antibody targeting the C terminus of calcitonin 3 available at Proteintech. It wasn't even on the market at the time because it hadn't been verified. We got a free sample from them, and lo and behold, it worked really beautifully under the right conditions. And that's actually the antibody we've been using throughout all of our studies up until this point. So this is showing that this protein actually exists endogenously in brown adipocytes. We have whole cell lysates and membrane preparations from control and a pool of a knockout mature brown adipocytes. And you can see here that in whole cell lysates, you can detect a band at around 25 kilodaltons that is lost in the pool. It's not 100% knockout because the pool is not 100% knockout. And in membrane preparations, you can also see an additional band around 40 kilodaltons that is also lost in the knockout. So we proved that this protein exists endogenously in brown adipocytes, and also that this antibody specifically targets this protein. Fortunately, this antibody also worked well for immunoprecipitation. We were able to pull down at least the smaller form, the smaller band here from brown adipocytes, and then using mass spec, we were able to identify one peptide that kind of matched the predicted sequence of the calcitonin 3 beta protein, proving that not only can we detect this protein, but we know the correct open reading frame and sequence. So the last part, before I kind of jump into the function of calcitonin 3 beta, is to note that the calcitonin 3 protein was previously described as a synapse organizing protein. It was thought to sit on the postsynaptic neuron membrane and interact through its N-terminal extracellular domain with alpha-neurexin proteins on the presynaptic nerve terminal. And this interaction was thought to drive formation of synapses. But as I mentioned a little bit earlier, this entire N-terminal extracellular domain is missing from the brown adipose tissue protein. And this C-terminal area really doesn't have any described function. So the brown adipose tissue protein really, prior to us and others studying it, didn't really have any clear function. The Tontinaz lab has a long history at this point of putting functions to uncharacterized proteins. And so one of the first steps we typically do is determining the subcellular localization of the protein. So here we generated a calcitonin 3 beta apex-2 C-terminal fusion protein. Apex-2 enables us to generate a product that's visible on electron microscopy. And here in a brown pre-adipocyte, you can see that these ER tubules are filled with this EM product that's generated by apex-2, which tells us that one, calcitonin 3 beta is is localized to the ER, and two, a little bit of topological information because the product is within the ER tubule, suggesting that the C-terminal end of this protein is actually within the ER lumen. Interestingly, once you treat the cells with oleic acid to induce lipid droplet formation, this protein completely localizes at the interface between the ER and the lipid droplets. You can see here that there's an ER tubule that's not associated with a lipid droplet, and it has been depleted of calcitonin 3 beta. It's only around the lipid droplet associated ER. Shown another way, in differentiated brown adipocytes, you can see again that these 4 different lipid droplets are surrounded by ER tubules that are positive for the apex-2 stain. And if you really zoom in carefully here, you can see a thin membrane surrounding this apex-2 EM product, suggesting that this protein is not necessarily at the surface of the lipid droplet. It is at the interface between the ER tubule and the lipid droplet. And another nice picture that we saw Essentially here, you've got an ER tubule that's coming off of a lipid droplet, and as soon as it kind of is separated from the lipid droplet, the calcitonin 3 beta signal disappears, showing that it's really specifically at the interface between these two organelles. To prove that these tubules are actually ER tubules, we took HeLa cells and treated them with lipoprotein deficient serum to deplete lipids, or with oleic acid to induce lipid droplet formation. And you can see here that calcitonin 3 beta overlaps nicely with an ER mark 661 beta in the absence of lipid droplets. However, when you treat with oleic acid to induce lipid droplet formation, now you see blue, in blue are the lipid droplets, and in yellow is the overlap between the ER and calcitonin 3 beta in these tubules that kind of wrap these lipid droplets up. And then we tried to determine which part of the protein was responsible for the lipid droplet targeting of this protein. So we generated a few different mutants. Here you have the full-length protein. We have a mutant where we've deleted the C-terminal region and are only leaving the 3 beta specific region. And we've also generated a mutant where the 3 beta specific domain is deleted, and we only have the region that's shared between the calcitonin 3 and calcitonin 3 beta proteins. And you can see here that the 3 beta specific domain is sufficient to go to lipid droplets, whereas in the absence of the 3 beta specific domain, you get intracellular localization that's not associated with lipid droplets, suggesting that the 3 beta specific domain is necessary and sufficient for localization to lipid droplets, which again makes perfect sense because the calcitonin 3 protein in the brain has not been shown to go to lipid droplets and is missing this entire domain here. Now, lipid droplet targeting generally occurs through two different mechanisms. There's one called the class 1 lipid droplet protein, which is characterized by the presence of hydrophobic hairpins. So these are essentially pairs of hydrophobic helices with a little hairpin turn in the middle, such that the helices do not traverse the entire lipid bilayer. And that's because lipid droplets are surrounded by a phospholipid monolayer, so that there's no hydrophilic surface on the other side of this membrane for proteins to interact with. So hairpins are a specific type of protein that like to go to lipid droplets. And so what they do is they sit on one leaflet of the ER membrane, and during lipid droplet biogenesis from the ER, they're able to kind of translocate onto the phospholipid monolayer of the lipid droplet. Class 2 lipid droplet proteins are characterized by amphipathic helices. These proteins essentially are thought to be disordered, and then in response to a packing defect in the phospholipid monolayer of the lipid droplet, forms an amphipathic helix with one side that's hydrophobic and one side that's hydrophilic, which enables interaction with both the cytosol and the lipid droplet. And we quickly realized that calcitonin 3 beta didn't seem to have any convincing amphipathic helices. And so what was most likely was that it formed hydrophobic hairpins. And computational structure prediction kind of supported this idea. Using Rosetta Fold, we were able to show that the novel calcitonin 3 beta specific region can be organized into 6 hydrophobic helices, which can be sort of paired into these 3 different hairpin-like structures. Hairpin 1 in blue, hairpin 2 in magenta, and hairpin 3 in yellow. And then we generated a mutant where we deleted the 3 hairpins and showed that this protein is no longer able to localize to lipid droplets. So to kind of summarize this first part, we've identified a novel brown adipocyte specific protein that assists in the formation of ER lipid droplet contacts and has a proposed structure that looks something like this, where you've got a transmembrane domain and a C-terminal domain that's shared with the calcitonin 3 protein. And it's connected by a cytosolic loop to a series of 3 hydrophobic hairpins that sit on the outer leaflet of the ER membrane. And then during lipid droplet formation, these this part of the protein is able to translocate onto the surface of the lipid droplets while this part stays anchored in the ER, which enables the formation of the contact site. So to determine the function of this protein, we generated global and adipocyte-specific calcitonin 3 beta knockout mice. So to generate the global mice, we took a CRISPR approach and essentially introduced an insertion in the novel calcitonin 3 beta exon to generate a knockout for the beta isoform, but not the brain protein. And you can see here that our knockout does not affect the 95 kilodalton calcitonin 3 protein, but is able to delete all forms of the calcitonin 3 beta protein in the brown adipose tissue. And for an adipocyte-specific knockout, we put LOXP sites around these last two exons here. And by crossing it with an adiponectin tree, we were able to delete it from brown and white adipose tissue, but not from the brain. First thing we noticed about these mice is that when you open them up after housing them at just room temperature, the brown adipose tissue looks different. You can see that they're larger and a little bit more pale colored, which typically suggests that they have increased deposition of lipids. And we measured triglycerides in the brown fat from both the adipocyte specific and the global knockout. And in both cases, you can see an increase in the amount of triglycerides per milligram of protein. Histologically, there is a really dramatic difference between the wild type and the knockout, with the knockout having a much more almost unilocular, but not quite, appearance with these really large lipid droplets in each cell, whereas the wild type had a kind of conventional multilocular phenotype. We also were able to show that the brown adipose tissue of these different models were a little bit defective by exposing the mice to a cold challenge. So in this experiment, we put the mice at a warm temperature of 30 degrees for about 2 weeks to kind of suppress any compensation mechanisms that were at play, and then immediately shifted them into the cold to kind of challenge their brown adipose tissue activity. And you can see here that in both models, over the course of 5 or 6 hours, the body temperature of the knockout mice is much lower than that of the wild type, suggesting that there's a moderate defect in the ability of the brown adipose tissue to generate heat. However, this was not due to differences in the expression of the thermogenic program. Here we have RNA sequencing data from brown adipose tissue before and after cold exposure. So here we have wild type knockout at 30 degrees, wild type knockout after 4 degrees. And you can see that, generally speaking, the genes that are induced or suppressed with cold exposure are not different between the wild type and the knockout, suggesting that the ability of the brown adipose tissue to sense the cold and turn on the genes that are important for thermogenesis was not impaired. And in longer term cold exposure, this is 4 degrees for 4 days, you can see that these canonical thermogenic genes are not changed in their expression pattern. What we did see is that the calcitonin 3 beta protein, when deleted, led to an increase in carbohydrate utilization versus lipid utilization. So this is measuring the respiratory exchange ratio over the course of 2 days. And a higher ratio, closer to 1, suggests that the mice are using more carbohydrates, and a lower ratio, closer to 0.7, indicates more lipid utilization. So in both models, we saw a bit of a trend towards an increase in carbohydrate utilization. Additionally, using PET imaging, we can show that the knockout mice actually take up more glucose in their brown adipose tissue under acute cold exposure. So you can see that visually here and quantified counts here. And this is specific to brown adipose tissue. You don't see it in any of these other kind of control tissues. This is interesting because, again, it shows that the brown adipose tissue is perfectly able to become activated in response to cold exposure. but there's some issue in terms of lipid versus carbohydrate utilization that's induced by knocking out this protein. So to investigate this a little bit more, we started to do gain of function studies. So here we took calcitonin 3 beta knockout brown adipocytes, and we expressed either M-cherry or calcitonin 3 beta tagged with an M-cherry in these cells. And notably, these proteins were kind of overexpressed during the differentiation process. And here, this is just a light microscopy image of differentiated brown adipocytes. You can see, multilocular, but got some pretty big lipid droplets here. But when you overexpress calcitonin 3 beta M-cherry, you really don't see very many large lipid droplets. In fact, these brown adipocytes are kind of studded with all of these little, pearly, small lipid droplets. That was really dramatic. It's something you can just, put your cell culture plate under a light microscope and see very clearly. To look at it a little bit more carefully, we did fluorescence microscopy. Here you can see two different cells that are expressing just a regular M-cherry. They've got nice large lipid droplets stained by BODIPI. And when you express calcitonin 3 beta M-cherry, you get just an accumulation of these tiny lipid droplets within the cell that are kind of intertwined with the red calcitonin 3 beta M-cherry signal. And here you can see the lipid droplet number is dramatically shifted towards the right in the presence of calcitonin 3 beta. And the mean lipid droplet area is also much smaller in the presence of calcitonin 3 beta. So this kind of suggests that the presence of 3 beta during the differentiation process, increases the multilocular phenotype of the brown adipocyte. This is a really interesting image that we took, where essentially within one view you can see multiple different adipocytes with different levels of 3 beta expression. Here you can see one where the 3 beta expression is essentially not visible, and the lipid droplets are nice and big. One with maybe more intermediate expression with more but smaller lipid droplets, and one with high expression where there are many more lipid droplets and they're all much smaller. So there also appears to be a bit of a dose dependency here in terms of the lipid droplet morphology. And while I was, finishing up my thesis, I saw these grapes at Trader Joe's, so grocery store in, I guess, in the US. And these champagne grapes were kind of next to these, regular red seedless grapes. And this kind of reminded me of the lipid droplets you see in a M-cherry expressing adipocyte versus a calcitonin 3 beta expressing adipocyte. So definitely it was on my mind a lot during those last several months. I just wanted to show some data to show that the adipocytes that are expressing 3 beta, they're not defective in any way. They differentiate normally. And in fact, they're more efficient at lipid utilization because they have all of these small lipid droplets. This is a seahorse study looking at mitochondrial respiration. And here in black and red, we have M-cherry versus calcitonin 3 beta M-cherry. And you can see that with uncoupling of the mitochondria, there's a much higher respiration rate in the calcitonin 3 beta M-cherry expressing cells versus the M-cherry. And when you introduce a inhibitor of fatty acid oxidation, adamoxir, this difference is minimized. When you normalize the protein, this difference is actually mostly gone, suggesting that there's an increase in mitochondrial respiration with 3 beta expression that is completely dependent on fatty acid oxidation. Shown a slightly different way, we isolated lipid droplets from M-cherry and calcitonin 3 beta expressing cells, and at 3 different speeds, getting different kind of populations of lipid droplets. And relative to the amount of triglyceride in the lipid droplets, you can see that the presence of 3 beta increases the amount of prolipolytic factors such as ATGL and CGI58. and also an ER marker, calnexin, you can see that there's just way more association between the ER and the lipid droplets in the presence of 3-beta. So taken together, this shows that it's not just a morphological difference, but this morphological difference in the lipid droplets translates to an increased efficiency of lipolysis and fatty acid oxidation in these cells. To show that we can do this in vivo, we took an interesting approach where we injected an AAV flex construct, which is essentially a Cre-dependent expression construct into the tail vein of these mice. They have to be knockout mice because the knockout has the Cre expressed in the adipocytes. And this allows us to achieve a adipocyte-specific re-expression of calcitonin 3-beta. And what we did afterwards is to expose the mice to cold for 4 days to kind of shrink all of the lipid droplets to their smallest state, and then put the mice at a warm temperature for a longer period to allow the lipid droplets to kind of grow and expand. And we wanted to see if expression of 3-beta was sufficient to kind of restrict the expansion of lipid droplets in brown adipose tissue. And what we saw is that indeed, when you overexpress 3-beta in the brown adipose tissue, there's a significant increase in the multilocular phenotype of 3-beta. So this protein is actually able to prevent, to some extent, the conversion of brown fat into a unilocular state at thermoneutral conditions. This is really exciting. We're kind of dissociating here the thermogenic activity of brown adipose tissue from its morphology. I think historically it's been hard to really dissociate an active brown fat from a multilocular brown fat, and whether or not those two were always dependent on each other, or whether there was a protein factor that was responsible for enforcing the multilocularity. And here we can show that indeed, even at thermoneutral conditions where brown fat is suppressed, that 3-beta is able to maintain a more multilocular phenotype. Interestingly, there are some human correlates here. This is a nice study out of Denmark, showing that in human multilocular versus unilocular fat, calcitonin 3 was identified as the #1 most significantly enriched gene in multilocular adipose tissue. And in our hands, we were able to show the same thing on tissue sections. So we have unilocular fat. Sorry, my cursor is missing. Yeah, I don't know where it went. But anyways, we have unilocular fat on the left side, and you don't see any expression of 3-beta. And on the right side, using an RNA scope probe, we're able to show that there are nuclei in multilocular adipocytes that express calcitonin 3-beta. And interestingly, you'll see a couple of unilocular adipocytes with the blue arrows where the nuclei are negative for calcitonin 3-beta, suggesting that the expression of this gene is also associated with multilocularity in humans. And lastly, we cloned human calcitonin 3-beta from one of these patients and show that it also goes to lipid droplets very strongly, suggesting that this is really an evolutionarily conserved function of this protein. So to summarize this second part, we did gain of function and loss of function studies for calcitonin 3-beta. We showed that gain of function was able to enforce multilocularity in cultured brown adipocytes and brown fat. It was able to increase the concentration of lipolytic factors on lipid droplets relative to the amount of triglyceride, and it was able to facilitate mitochondrial fatty acid oxidation. In loss of function, the brown adipose tissue had increased lipid content and also a more unilocular lipid droplet phenotype. The mice were moderately more susceptible to a cold challenge, and there was a compensatory shift in substrate utilization from lipid towards carbohydrate, suggesting that there was a bit of a defect in the lipid utilization in the brown adipose tissue. And as a little bit of a bonus, we found some human associations where in previous studies, it was shown to be the most significantly enriched gene in human multilocular versus unilocular fat. And we were able to show that it's expressed specifically in the multilocular adipocytes within those kind of heterogeneous sections. The last part of our story is essentially the mechanism. How does calcitonin 3 beta enforce a multilocular phenotype? And how we tackled this problem was first to identify the interactome of this protein. We took two approaches. We used apex proximity labeling and also flag immunoprecipitation, and took the overlap between these two data sets to identify high confidence interacting proteins. On the right-hand side, you can see that this interactome was significantly enriched for lipid droplet and ER-associated proteins. And among the lipid droplet proteins, we honed in on a protein called SIDE-C for reasons I'll discuss in a minute. Essentially, SIDE-C has been shown to be responsible for the unilocular phenotype of a white adipocyte. So if you knock out SIDE-C, the white adipose tissue becomes highly multilocular, as you can see in the H&E stains below. So this was really a natural target for us to look at in terms of the mechanism. Shoot, I don't have my cursor, so I can't start this video. Let me try, oh, here we go. Ooh. Here we go, okay. This is a video showing the ability of side overexpression to facilitate the coalescence of lipid droplets over time. As you can see, you started with a bunch of small lipid droplets, and over time, the expression of the side protein enables the lipid droplets to coalesce into larger ones. How do they accomplish this feat? These are lipid droplet associated proteins that allow for docking between two lipid droplets. And over time, there's net lipid transfer from the smaller one into the larger one due to differences in surface tension. And over time, this results in the fusion of two lipid droplets into one larger one. Interestingly, in this paper, they actually kind of predicted the existence of 3 beta without knowing it. They write that lipid droplet enlargement in white adipocytes would occur until a unilocular phenotype is achieved, whereas in brown adipocytes expressing side proteins, this process would be stopped at the multilocular stage. They hypothesized that would be due to post-translational modifications or other factors. We think that calcitonin 3 beta is one of the key factors at play here. So first, we showed in different cell types that there's co-localization between calcitonin 3 beta and side proteins at the interface between lipid droplets. Here you can see in yellow, a nice co-localization at the interface between two different lipid droplets. And then we measured the ability of the lipid droplets to transfer lipids between them in the presence of calcitonin 3 beta. So I'll just walk you through this experiment as it's a pretty important one for our paper. So this is essentially a FRAP experiment, a fluorescence recovery after photobleaching. What we're doing here is photobleaching one of the lipid droplets and watching it fill up with the lipids from the adjacent lipid droplet over time. So here you can see that in the presence of 3 beta, when it's been photobleached, there's minimal recovery of the fluorescence over time, whereas in the condition minus calcitonin 3 beta, there's more recovery. And you can kind of repeat this photobleaching process again. You've bleached, there's more recovery in this condition lacking calcitonin 3 beta. And a third time, there's a little bit of recovery here, but it stays pretty dark in the 3 beta condition. This is nicely quantified here, where I guess bleaching, recovery, bleaching, recovery, bleaching, recovery, you see with SIDE-C. But in the presence of calcitonin 3 beta, this recovery is really flatlined, suggesting that through a yet to be determined mechanism, the expression of calcitonin 3 beta prevents the ability of sides to kind of transfer a lipid between the lipid droplets, and thereby prevents them from fusing together and forming a more unilocular phenotype. This is a video of this experiment. Again, you can see each bleaching and recovery in the SIDE-C expressing group, while as the calcitonin 3 beta expression, this lipid droplet stays dark for most of the experiment. And quantified nicely here, you have side A and side C, both of them increase lipid exchange between the lipid droplets. And then if you express 3 beta, this activity is almost completely suppressed. So to kind of summarize this last part and kind of the overall story, we kind of looked at calcitonin 3 beta knockout, brown adipocytes, wild type, and overexpression. And in these 3 different conditions, we see 3 different lipid droplet phenotypes. We see a more exaggerated unilocular phenotype, a more exaggerated multilocular phenotype, and kind of your standard brown adipocyte phenotype. And we think that this is in large part due to the activity of 3 beta inside. So in the absence of 3 beta, there's more net lipid transfer into the larger lipid droplet, which allows for formation of more unilocular adipocytes. In the presence of 3 beta, there's some inhibition of side lipid transfer activity through a mechanism that we're still working out. And so when you overexpress 3 beta, there's really minimal lipid transfer between these small lipid droplets, leading to the accumulation of this kind of multilocular lipid droplet phenotype. And we think that this multilocular phenotype favors triglyceride utilization, whereas in the knockout, they're less able to use lipids efficiently, and so kind of switch to more of a glucose utilization. So this is kind of the end of the talk. I just want to give some acknowledgements here. And I was also asked to talk a little bit about my graduate school experience, which I think was a little bit unique in some ways. So I'd like to thank the Tontonos Lab. This was really a group effort. Peter and different postdocs and graduate students in the lab. We have a ton of collaborators, most importantly, Peng Li's lab at Tsinghua University. They're really the experts in side proteins and did the majority of the functional studies on side. My funding sources, and in terms of my graduate school experience, I think it was a little bit unique because I started working on this project in 2017, so 5.5 years ago. And about a couple of years into my PhD, another paper was published on the calcitonin 3 beta protein. I won't go into too much detail, but you can, if you search calcitonin 3 beta, you'll find a couple of different papers on it. And long story short, obviously I was devastated. Being scooped is never a good feeling. But I think, we put our heads down and really tried to get to the bottom of the story and came up with some maybe different findings than the initial paper. And that kind of gave us, a new angle to the story that kind of excited people in the lipid droplet field, people in the adipose field, and also our reviewers and editors. And so, this paper ended up being published in Nature, and I have to give a lot of credit to Nature for being willing to kind of explore this story with us and not kind of ignore it based on the fact that there was another paper published. And so, yeah, I think the lesson that I took away from this is that, Peter always says this, but, if you put your head down and do good science, that it'll be recognized. And, just because one paper has been published about something doesn't mean that it's the end of the story. So with that, I will take any questions.
Awesome. Thank you so much, Dr. Qian, for sharing your really interesting work and also your story. We're always all afraid to get scooped, and especially in, when you're in graduate school. And so it's really nice to kind of hear your story and see the happy ending. So before we start the Q&A session, I just wanted to share a few more slides highlighting some products that might be interesting for our audience today. So I just wanted to mention that Proteintech offers top-sided antibodies for brown adipose tissue research. So we have antibodies for mitochondria and antibodies that are markers for brown adipose tissue as well as white adipose tissue. And then if you're interested, we also have a list of the antibodies that Dr. Qian used in his paper. I also want to highlight a newer addition to our portfolio, our flexible kits. So flexible is a novel antibody labeling kit that uses an affinity linker to conjugate fluorochromes, enzymes, and molecules in any buffer condition. It's compatible with any primary antibody, so it doesn't matter where you get your antibodies from. It can be from any supplier. And you can use the kit to label as little as zero 0.5 micrograms of antibody for individual experiments, and your labeled antibodies are ready to go in 10 minutes. So one of the main benefits of using our flexible antibody labeling kits is that it allows you to multiplex antibodies from the same species. So right now we have kits for antibodies against rabbit IgG, mouse IgG1, and mouse IgG2A in various different colors. Also, as an attendee of today's webinar, you're entitled to receive a free sample of any of our unconjugated antibodies. You will receive a follow-up email that will have a link with a form where you can request your free antibody. Also, we'll be sending certificates of attendance to verified attendees. So with that, we can go ahead and begin the Q&A session. After this Q&A session, if you have any other follow-up questions, please feel free to email us at events@ptglab.com, or you can email Dr. Qian directly at kqian@mednet.ucla.edu. So with that, I'll go ahead and start the Q&A session. Okay. So here we have a question saying, in the loss of function for calcitonin 3 beta, as they are utilizing more glucose, do you think this will be helpful for hyperglycemia? And gain of function, do you think this will be helpful more for dyslipidemia?
Yeah, I think that's a great question. There are quite a few papers showing kind of impairments in lipid utilization in brown fat, leading to kind of an increase in glucose consumption, and that it might have some protective effects against hyperglycemia, at least in cold conditions. That isn't something that we've tried yet or that we've resolved yet. I think that's going to require some more experiments. But we would essentially try to do like a glucose tolerance test in the cold and see if there's any difference between the two conditions. But I certainly think that's a possibility.
We have someone here who really liked your videos that you included and was wondering how long you imaged the cells.
Yeah, I'm assuming we're referring to the FRAP images, which weren't done by me, but by our collaborator in Tsinghua University. If you look at our paper, you can see that each cycle was done over, I think, 60 to 90 seconds. If you're referring to the kind of longer video with the coalescence of the lipid droplets in the presence of Psi, that was from a published paper, Barneda et al. In eLife 2015. So you can find that paper.
And we have one more question so far that's asking, in knockout adipocytes, glucose consumption is increased. Does it change the pH?
I think by pH, you might be referring to like acidity generated from glycolysis. That isn't something that we've directly measured, but that would be something interesting to look at. I think part of the challenge of, studying glycolysis would be that, the compensation really happens in an in vivo setting, right? Like the mice need an alternative way to generate heat, so they switch to glucose utilization. But in vitro setting, where we typically do seahorse experiments and measure glycolysis, there's really not a need to make that kind of substrate switch because the cells are kind of existing on their own. They don't need thermogenesis to survive. So we would have to, I think, find a good way to study that in vivo.
Thanks. So we have another question that came in. In knockout adipocytes, what happens to glucose importer? Does increased glucose level inhibit the lipid droplet fusion?
Another great question. We haven't specifically looked at that, but there are a few other papers out there where they've kind of, inhibited different parts of the lipolysis process in brown adipocytes, and they consistently see increased, GLUT4 expression on the plasma membrane, increased glucose uptake. We kind of just looked at more of the functional aspect, but I would suspect that there's more GLUT4 expression on the plasma membrane.
Right. So we have a question that came in the Q&A section. So someone says, thank you for referencing their work. Did you find any difference between interactions, physical or functional, of calcitonin 3 beta and CIDEA versus CIDEC?
Yeah, thanks so much for attending. I saw your name in the attendance list, and I was like, "This is, your paper was really critical in kind of understanding our data and story." So in the supplement of our paper, we do some experiments looking at interaction between calcitonin 3 beta and side A versus side C. We see that grossly speaking, that 3 beta seems to interact better with side A and a little bit less with side C. In terms of functional assays, side A generally seems to have a lower lipid transfer rate than side C, at least according to studies that Peng Li has done. And both are able to be suppressed by 3 beta. So at least the ability to suppress lipid transfer is pretty consistent between the two, but there does seem to be a little bit of a stronger physical association with side A.
Great. Well, thank you. I think that's all the time we have for questions. I just want to thank everyone for attending. And again, thank you, Dr. Qian, for sharing your work and also your experiences in grad school. Again, if you have any follow-up questions, please feel free to email us at events@ptglab.com or email Dr. Qian directly. We'll include his email in the follow-up email as well so that you have access to that. So with that, I'll go ahead and end the webinar and hope everyone has a good rest of your day. Thanks.